My name is Raquel Barrett. I'm with Genome Prairie. I'm a project manager for Genome 360 in Manitoba. I'm fairly new to Genome Prairie. I've only been here 3 months, but I've been around bioscience and life science industry for several years now. We're coming to you from downtown Winnipeg. And oh, I should have put a background on. Too late. I've got the Richardson building looming over me here. I can see the grain exchange. So this is definitely grain center, agricultural center. But this place has also been a meeting place for thousands of years. It's now Treaty 1 territory. And it is the traditional lands of the Anishinaabe, the Cree, the Oji-Cree, the Dakota, Dene, and the birthplace of the Métis Nation. So we're very grateful for the use of this land and respect the treaties. So actually, before I start talking about SeekNet, a few housekeeping things. You're all on mute. We're going to save questions till the end. And if you want to ask a question, you can put it in the Q&A, or you can, I think you can raise your hand, and then I can unmute you. And of course, we are recording, so hopefully you're all okay with that. So I'm just going to share my screen. And hopefully you can see that. And the controls are in the way, so I can't, it won't let, that's annoying. Okay, an unanticipated problem there. I'm gonna unshare. No, that doesn't help. Okay. Yeah, I know it's a bit frustrating. I'm gonna go back. I'm just gonna show you the show like this. Unfortunately, I can't make it full screen for some reason because all of these controls are in the way across the top, which I hadn't thought about. But I think you're going to get the general idea of, I think you can see entirely the actual slideshow. Okay, so the SeekNet project, our goal is to help the province adopt genomics technologies in the agricultural sector specifically. And we really want to reduce Manitobans' reliance on out-of-province genomics services. We've done this by building capacity through the use of next-generation sequencing equipment that we've placed in a few different locations. And we're also encouraging the training of highly qualified personnel. SeekNet is a Genome 360 project supported by Agriculture Canada through the Canadian Agricultural Partnership Program and Manitoba Agriculture and Resource Development, as well as a Genome Prairie. So we're very grateful for that support. The way in which you could collaborate with us is if you're working in agriculture, you would need to be working in agriculture, and you're currently sending samples outside the province for analysis. Or even if you're planning a research program that's going to require genomics analysis, or if you're currently doing testing, but you're thinking maybe you could do the testing by a genomics method rather than your current method. Those are all great reasons to contact us, or even if you just want to find out more information. But if you're in one of those situations, we could arrange to have one of our partners to do sequencing for you or other testing. And we will just find some way to actually make that work for you. So I am the Genome 360 project manager in Manitoba. My counterpart in Saskatchewan is Rahwa Osman. The SeekNet project is only in Manitoba, but you're certainly welcome to contact Rahwa if you're in Saskatchewan and learn about some of the opportunities available as Genome 360 there. Okay. Now this is awkward. Okay. So now I want to introduce our two speakers. Well, our first speaker will be Dr. Leanne Murphy. Leanne is the Vice President of Murphy et al. Incorporated, an agricultural research group that creates value for clients through the execution of scientifically and agronomically sound, confidential and comprehensive agricultural research programs. For nearly 20 years, she's been conducting variety, efficacy, disease, and other agronomic trials for private industry, commodity groups, and government agencies. For the past 6 years, Dr. Murphy has also been the CEO of Pest Surveillance Initiative, a grower lab focused on validating and deploying DNA-based assays to detect, characterize, and quantify soil-borne, aerial, and insect-vectored cropped pathogens. Dr. Murphy has a PhD in plant science from University of Manitoba and is a registered professional agrologist. Dr. Murphy has previously held positions such as Managing Director at DivSeek International, Executive Director of the Manitoba Agri-Health Research Network, and President of NUEADS. She's going to talk to us today about the evolution of crop biosurveillance as an opportunity for DNA-based testing. And I'm just going to make it so that okay. There you go.
Are you able to see my screen? Yeah.
It says you've started screen sharing, but I just see blackness.
Got it. You've released on your end?
Yeah, it says you've started sharing.
How about now?
Ah, there you go.
Okay. Super. Well, thanks everyone. I'll assume I'm unmuted. Pleasure to be here and to talk about Pest Surveillance Initiative, or PSI. About 20 slides just to introduce and share some of, as Raquel said, the goal of today is how we're actually using genomics information, to help growers in our case. Advanced screen is not working. Okay. A little lag here. So I'll just a quick overview of what I'm going to talk about, a little bit about PSI, how we're using DNA-based testing strategies to help growers, and then some of the projects that are currently in our pipeline. And currently in our pipeline in our world means underway and about to be deployed. So first off, what is Pest Surveillance Initiative, or PSI? As said, this is a grower-led, and that's the unique part of what we do. The Manitoba Canola Growers Association owns and operates PSI. So it's growers investing for grower information. As Raquel said, we identify, quantify, and track risks to successful crop production. And our focus is really on what pests are in the Manitoba environment that could impact yield. Operationally, that means that what we do is we scour the literature in the world for research discoveries that we can take off the lab bench and deploy into practice. Of course, before we can do that, we need to optimize and validate all the tests. Then we also like to keep a DNA and sample archive so that we can dip back in when new tests either are released or if we find we need to tweak something. And while we test commercially, our goal is really to offer testing where it's not widely available commercially. So these would be one-off assays or assays that are very unique to Manitoba grower situation. And then just a little bit of the historical. We opened in the fall of 2014 in response to the first detection of Clubroot in Manitoba. We provided the DNA-based testing capacity and still do for the province to do the DNA quantification. As of now, and it's growing every day, more than 2,000 physical and DNA samples that we keep in our archive. We tend to keep positive samples only, or else we would have way more than that. The facility itself, 1,300 square feet, so quite compact. We combine a genomic ID section, so PCR, qPCR. We use the LAMP assay and as part of our participation with Genome 360 and the SeekNet project, we have one of the iSeeks at PSI. We do a lot of phenotyping as well, so what I would call traditional plant pathology, single spore isolates that we generate from field material, differential media, that type of thing. We also have access to a field lab, so we can collect field samples. And with support from the CAP program, we were able to put in a small plant growth cabinet, again, to generate some of our material. The technologies we really focus on are on platforms. So it's surveillance and sampling, characterizing the pathogens that we're looking at. And then again, a field nursery evaluation system where we can look at natural as well as artificial infection. So this, if it doesn't like too bad, is a quick look inside. So PSI is separated into 3 sections. We have our intake spot. We have this part, which is our clean lab with all the equipment. And then we have an office space. You can see qPCR there. It's a robotic sampler. Lots of freezers. I don't know that there's a DNA lab out there that doesn't have freezers everywhere because sample quality is important. And you can see some of the plates and traditional pathology tools that we use. They're really compact and really focused. Oops. Okay. One of the important things when we talk about how do we help growers is understanding how that information gets to them. And as I mentioned, Manitoba Canola Growers, has invested in the concept of DNA-based testing for Manitoba. And one of the projects that we do on their behalf is called Get Tested. And this is a really innovative program where MCGA covers the cost of testing for all their member fields for an increasing array of pathogens, that or pests that would affect crop production. So blackleg race, and I'll explain some of these later on. Verticillium stripe, which is a new pathogen. We're trying to understand the economic scope. Clubroot, as I mentioned, glyphosate-resistant kochia, and just this year, amaranth species ID. And again, I'll cover that a little bit more. The Get Tested program really continues to evolve in response to what the growers are telling us they needed. Initially, it was, we'll do Clubroot testing for you. And they said, well, that's not good enough. So what else can you do for us? So we covered all the tests for all the fields. And then in this past year, and that captures what's evolving in the field, is that agronomists could submit samples on behalf of their members. And that really helps to close the link between identification of the problem, finding a solution, and doing that quickly. So in 2021, 9 different agronomy service companies worked with MCGA members to submit their samples. And as I said, MCGA is very encouraging of any new assays that we develop, that we deploy as soon as they're validated. Those aren't just restricted to canola pathogens, because of course, every grower is growing canola 1 in 3 or 1 in 4 years. And what happens on the land in those interim years can impact their canola production. So again, very holistic approach. So I mentioned about Clubroot, and this is really where PSI got started. Clubroot can cause significant yield loss in susceptible varieties. You can see these are Clubroots up here. This is a lab sample, pretty clean, but all that means that the plant is not thriving. The challenge with Clubroot is that it is a soil-borne disease. So we get samples of soil. We have to size reduce those very carefully, very manually, because our focus is on low limit of detection of spores that are found in the soil. Anybody who's worked with soil knows it's a very challenging matrix to begin with. Our focus on low-level detection makes it just crucial for our program that we take a lot of care in sample preparation to ensure that we're actually capturing the spores that may be present in the soil. A little bit of facts and figures. Half-life of spores is 4 years. Could be more than that depending on conditions. So if you had millions of spores, you can see that the problem will persist as the half-life only goes down in those interim years. We can see infection at about 100,000 spores per gram of soil in the field, which again is not a lot of spore material to start seeing some damage. And again, because of Manitoba's Clubroot situation, which compared to some of the other provinces or other regions in the globe, is not at the level that of huge impact, we want to keep it that way. So that our focus continues on low-level detection so that growers have the opportunity to change management to make sure that we don't have to manage Clubroot, but we can work to try and prevent or limit its presence. Our low limit detection is about 800 spores per gram of soil. So again, very, very refined, level of detection in soil. But we don't just stop at what does the DNA tell us about the spores? Because again, remember, the whole premise behind DNA testing is that this aids growers. So when we provide a result back to the growers, we look at also including some interpretation and then management guidance. And this is really a hallmark of all of our tests. So this example, this would be a typical report that we send out. We would drop in whatever the spores per gram of soil number is. It would be against a grid that shows what the different impact of that level of spores in the soil sample is. And more importantly, what the grower can do to manage around that information. We're lucky that we haven't had to go too far above the 80,000. We've had million spore counts in Manitoba, but the vast majority are in this low area, which is, again, our goal of trying to keep it there. I also mentioned, amaranth. So that's the pigweed family for folks that, want to think of them that way. This is an invasive weed species that I guess it was only a matter of time till we were going to see in Manitoba. So part of our goal was to be ready with an assay when most of our pathogens move up from the state. So once Minnesota and North Dakota started getting some of these really, yield devastating invasive species, again, it was only a matter of time. The challenge that we've got, and I'll explain a little bit about amaranth and then how we test for it. There's probably 5 common amaranth species. There's probably more, but we'll call it 5. 3 of them in Manitoba have been detected now are tier 1 noxious weeds, which means under that, the Weeds Act, they must be destroyed. The other ones are not tier 1 and don't need to be destroyed. The challenge is that we want to identify these as early as possible in the field, and certainly before there's seed production, because once an amaranth produces seed, it's viable, it stays in the soil, and there could be thousands of them. So preventing seed set is really the goal of all the management. But if you can't identify it, then you don't know what you're managing. They've tried a lot of visual IDs, and this is an example at the vegetative stage. This is not Manitoba material, because we'll get to the whole biotype thing in a bit as well. So this is Palmer amaranth, female leaf, because it has male and female plants. That's a tier 1 noxious. This is tall water hemp, tier 1 noxious. This is Powell, which we have in Manitoba, not tier 1 noxious. And this is red root pigweed, almost an endemic common weed. You have to be very skilled and hope that you get a consistent expression of these different leaf types and petiole lengths to use that as an ID. Because of the tier 1 noxious category, we want to be sure before we're going into fields or having growers, tear up patches of a weed, which is why we worked with the University of Illinois to bring in a molecular testing system so that we can identify at least the tier 1s with a yes, no. And again, this is just the start of our work with amaranth. It's a green tissue test, so it's on the leaf. And so the results get back to the grower in time for them to take a management decision. And you can see very simple yes, no. In our test samples, we encourage sampling across a patch so that we get a good feel for what's in there. And we'll go yes, no based on a molecular marker test. So again, a great example of timely information that really has an economic impact for growers, either positively because it's not a tier 1 noxious weed that needs to be destroyed, or conversely, it must be managed quickly. The next example I'm going to use is blackleg of canola. Again, blackleg, in canola is a disease that we've been dealing with, we'll continue to deal with. Yield losses of up to 50% recorded in some fields, so serious economic impact on canola growing. There are 2 types of blackleg resistance in our current varieties, major gene, which again is specific to race, and then a host of minor genes. It's the major gene resistance that is currently losing some of its effectiveness in our varieties. Of course, if we had just a simple yes, no, you've got, this, race in your field, that would be great, except the races exist as a population in the field, which hugely impacts the number of assay work or the kind of work that we have to do in order to understand what's in the field. And then, of course, because nature loves to play tricks, especially with pathologists, the races will actually interact with each other so that they will suppress the activity of one that you think should be there and providing protection. So just understanding the genotype without understanding the phenotype doesn't give the grower the answer that they need. So Agriculture Canada developed a series, a KASP-based system. That's one of the systems where we basically run the virulence and virulence markers against the samples. We try to get up to 8 symptomatic stems. Again, this is not just a survey. We want to identify the races, so we want symptomatic material. We will look at that relationship and then come up with a score basically for that set of submitted sample. So this would represent the genotype. When we report to growers, as I said, the genotype's interesting, but unfortunately it's the phenotype, or fortunately it's the phenotype where we see the we see in the field. So that's what's really important from a grower perspective. Based on the research that's been done by all the different science groups looking at blackleg and the pathogen, there's an estimation of phenotype that takes care of some of those interactions or takes them into account. So we get a phenotype that we estimate, and then we couple that with a recommendation based on what we know about the resistance change groups. So all of these correspond to different resistance types in a particular variety. And so we're very comfortable giving a recommendation based on what we know about the interaction. As those interactions change or update, as the race mix, or the markers develop, we will continue to evolve that assay. Why this is important for growers is that canola varieties are assigned a resistance rating. They're called the R genes. And so each bag has the opportunity, each bag of seed has the opportunity to be labeled. It's a voluntary system, so not all varieties have that. So again, we encourage the grower to use this information to talk to their seed supplier, about if I had this result and I should not be having this resistance group, what is the best choice of variety for us? You'll also notice Verticillium longiforme appears on this report. And then again, is one of those, by being proactive and ready, we were able to address a pathogen that we're seeing in Manitoba. Still not sure the economic impact. We'll wait for the science on that. But every stem that comes in for blackleg examination, we automatically and routinely look for Verticillium. And why this is important is that they will co-present, so we can have blackleg and Verticillium on the same stem. The grower just wants to know, what is this? Unfortunately, with Verticillium, we don't have a control answer. It is soil-borne, so again, it's probably that, lengthen the rotation, reduce the spore load, but we don't know. So, but we continue to monitor those so that we have an idea of how endemic this pathogen is in Manitoba. Okay, so now that's kind of what we've done, and now we'll build on that platform as we look into some of the future tasks or future opportunities around DNA testing and crop biosurveillance. For us, sequencing really has opened the door to some of the tools that we can do ourselves versus waiting for others to discover them, publish them, we bring it in, we validate. This allows us to go more directly to rapidly identifying either new races or pathotypes, or the range of pathotypes of existing, biotypes of existing pathogens. For us, this is a huge stepwise improvement in traditional bioassays. Anybody who's done a bioassay for pathogens or for weeds will know that they can be subjective, they're very laborious, and they consume a huge amount of space because you're basically growing out plants in replicate and then damaging them only to score them. So we view the sequencing as just a step change of improvement. It allows us to leverage globally reported markers and see if they actually make sense for our local collections. It also, when we find something that doesn't fit what we expected, allows us to say maybe that is something of either novel or it's emerging, and again, complete the circle with the science groups. And then for target-site herbicide resistance, which is increasingly seen across our weed spectrum, we have the potential to stack assays because if it's target site, it's usually a single gene, so we can work with one sample and multiple targets. For us, really, by identifying the races and biotypes that are present in Manitoba, that's the way that we're going to help growers stay on top of them so that we either have a good solution in place or we have a management technique until there's a genetic solution through a variety that allows them to manage that pest pressure. Coupled with that is an opportunity around what we're calling crop pest phenotyping. Now, you may be familiar with phenotyping, that a lot of it's being done on the crop side. So understanding, basically in my mind, how the environment and the genotype express what we're seeing in the field. So we have established what we're calling our crop pest phenotyping unit. That's really going to start to take the samples that we're already collecting, couple it with the DNA information that we've got, add it to the sequencing that we're doing, and really get a full picture of that. And this allows us to leverage, DNA technology in our existing platforms into the new economy of digital infrastructure. I know, and this is recognized it's the start. So it really involves a digital imaging system because we want to understand, the 3D morphology of whether it's spores or our cultures or our tissues. Think of that amaranth species. There are parts of the plant not easily seen with the naked eye that might be the difference between the species. So we want to understand that better. This will allow us then to have a sample bank and now an image bank to eventually field deploy some of these digital tools. Our ultimate goal is to do as rapid a detection in the field as we can. If we have to collect a sample, send it into the lab, analyze and wait for the result, that just takes time. If there was a quick yes, no, even in the field, then the yeses would get all the extra work and the no's, would self-identify. So that's kind of the goal, is to get to in-field deploy rapid tools. And then obviously generating, just large volumes of data, so workstations. So for us, the two key pieces of digital infrastructure that we've invested in is an imaging system as well as this high capacity workstation that will allow us again to really bring everything all together into one database. SeekNet is the SeekNet, initiative, the SeekNet community, as I call it, coming out of the Genome 360 has really been important for us in a couple of ways. Obviously, increasing the speed of Pest ID is what we're interested in, and then access to that sequencing capacity, the right size for our size of lab is exactly what this, the iSeek, for example, from Illumina, that's the platform that we're choosing to use. But I think it's also important to recognize, I guess, an industrial end user of technology. There's things that we also need out of the system, and that's what the SeekNet initiative has allowed us to do. Things like this, which I call peer level, exchange of information around technology, connecting with trained staff, whether they be in a university environment or in a company, on how to operate or tweak some of the new equipment and vice versa. But I think most important for us is that when we find a challenge in the field that we can't identify or haven't seen, that we have a conduit really to feed that back into the research community for continued investigation. Because that's really only how growers will ultimately benefit from finding something. It's like, well, you found it, but now what? And we try to answer the now what as quickly as we can, not just the here it is, in 5 years, I'll tell you what it means. I think we expect more, and I think through the examples I've shared, I think it can be delivered. A little bit of sort of my crystal ball, if I had one, as I mentioned, moving more towards field level assays is really, to me, that's what everything we do is geared for. We look at a lot of stuff, we say, I'm not sure that makes sense. We'll try the tool. If the tool doesn't work, we move on. We're not wed to any one platform. I really think that this race level ID versus species with DNA-based testing should become the norm. We can do better. We should do better. Whether it is, the cereal pathogen like Sean's going to talk about, or what we're seeing in canola or peas, pick a crop. We have the tool. We should be able to use that information. We also would like to see testing evolve to what I call a whole farm approach versus a crop pathogen silo. If the races of a pathogen are interacting with each other, what is the impact of other soil microorganisms? What is the role of crop rotation? And we know it manages disease, but what is it doing with the pathogen mix? Maybe we can get rid of the more virulent, strains by doing different things. Don't know unless we take a whole farm approach. And then obviously integrating DNA testing. If somebody's driving a piece of equipment around with sensors in a field, collecting some DNA should be something that could become the norm, so that we're getting, again, a full picture of what's happening instead of just a visual. So with that, the future is always bright and sunny, regardless of what it looks like outside. And I thank you for the opportunity to share a little bit about what we do, and more importantly, why DNA is really core to what we're trying to achieve.
Thanks, Leanne. I think I'm kind of glinting here, but hopefully you can still see me. Okay, so next we're going to have Dr. Sean Walkowiak. He's a research scientist and program manager for microbiology and grain genomics at the Canadian Grain Commission of the Government of Canada, and an adjunct professor at the University of Manitoba. He has over 10 years of experience performing applied research on wheat and its diseases, with a specialization in genomics, wheat breeding, and fusarium head blight. A main focus of his research includes the development of DNA testing methods for microbe and crop variety identification and monitoring, This program also performs annual monitoring and surveillance, including fusarium surveys for wheat. Dr. Walkowiak's research has been published in high-quality research journals, including Science, PNAS, Nature, and Nature Genetics. His lab is located at the Richardson Center for Functional Foods and Nutraceuticals at the University of Manitoba and is certified for level 2 human and plant pathogens and equipped with the latest tools in microbiology, genetics, and genomics. So he's got the great title for his talk today, Applying Genomics to Cereals and Cereal Killers. Thanks, Sean.
There you go. Hopefully that unmuted me so that you guys can hear me okay. Yeah, so thanks for the introduction and the invitation to connect with everybody here today. I hope this leads to some nice discussions, and it's a hard act to follow with Leanne providing such a nice overview of all the different work that she's done in canola. But I'm going to switch gears and talk a little bit more about cereals and cereal killers with that punny serial killers title. I like to use that one. So before I get started on the nuts and bolts of cereals and cereal killers, I'm going to talk a bit about the Grain Commission. So the Grain Commission is a science-based government department, and we provide support for producers in the grain industry to help maintain the grain grading system and ensure the dependability of Canadian grain, its quality, its safety, which ties into my interest in the microbes, and trying to secure, the future of the market, both domestically and internationally. And there's many different divisions within the Grain Commission, so there's lots of moving parts. And I'm going to be focusing on the Grain Research Lab, which is our research division, which provides support for many of the other components within the Grain Commission, including our inspectors and a lot of the standards in grain grading system, as I mentioned. So within the Grain Research Laboratory, there's several different programs that focus on crop research. So those would be at the bottom here. We have a bread and durum wheat program. We have programs that look at milling, malting, and barley and other grains. Recently, we've added canary seed to that list of grains that we look at. We look at oil seeds, so soybean, canola, mustard, flax. We look at pulses, so the lentils, beans, peas. So we cover all the different official grains within Canada. But we also do technology research. So we have more chemistry labs that do organic and element analysis. So a lot of analysis of pesticide residues and heavy metals and these types of things on grains. And then we have a biotechnology and variety monitoring group. So they would be doing DNA-based testing for identifying varieties in grain samples as part of requirements for export or to meet the needs of individual clients. So for example, malters often like there to be a certain variety purity when they do their malting. So we have, digital PCR tests for assessing barley, and we have an open array system which runs a panel of markers to identify varieties for wheat, for example, as a requirement for many of those clients. And many of those tests are developed in my program, which is the Microbiology and Grain Genomics program. And we do DNA-based testing of microbes, and we also do sequencing to develop DNA tests for both varieties and for the microbes to better identify them and monitor different targets of interest. When we look at what types of samples we're trying to do this monitoring in, we work really closely with cargo samples that are going to people that are trying to import Canadian grain. They often have requirements to do different types of testing to ensure that they're within certain expectations. We also do offer services for producers. So we have the Harvest Sample Program, which is a voluntary program that producers can participate in. And we basically just send you an envelope, you fill it up with grain and you send it to us, and then we'll give you an unofficial grade of that grain, as well as some important information about the microbes that might be in there, such as if there's fusarium damage or ergot damage or some of these other things to think about. We also do requests for service analysis. So for example, in my lab, we often provide some additional support for DNA-based testing labs that are in industry to give a second opinion as to what something might be, if there's something that they're having trouble with. Or we also share different methods between ourselves and the seed testing companies, so that we're all sort of best equipped to be able to have the latest tools for testing for different things for producers. The Grain Research Lab also analyzes the plant breeder lines from the private and public breeders as part of the variety registration system. So we provide that sort of independent support as part of variety registration. So my program focuses more closely on the genomics aspect. So to take a few steps back when we're looking at DNA, so DNA is the code of life. It's what encodes the genes or RNAs, which then get translated into proteins, which are the functional units in all living things. And it's the differences in these DNA that give rise to trait differences in crops, or race specificity in the case of Leanne's avirulence genes. So these are the types of things that we're interested in capturing, and the best way to capture that is through genomics. And there have been a few challenges throughout the years in trying to capture genome information. In the early 1990s and early 2000s, the cost was high and the throughput was low. To sequence a human genome, it would have been around $100 million at the time when they started sequencing the human genome. But for 600 big ones, you can get a human genome today. So times have changed. The accessibility to sequencing information is increasing. The costs are going down, and it's now reasonable to sequence organisms within individual laboratories, as opposed to large groups of individuals that are required to sort of interact together to get something done. The tools are now available in single labs. First levels of sequencing were usually done in microbes, so bacteria and fungi. And only recently we've been getting genomes for higher organisms like wheat, because the genome size of wheat, for example, is very large and it's repetitive, so it's much more complex. So it requires a lot more these technology changes to enable the access to those genome resources. So to give you an idea in the change of cost, it's the Fusarium genome was sequenced in 2002, and now you can sequence, a few hundred of them at once fairly easily. But at the time, not that long ago, that was a huge accomplishment. But the price and throughput, like I said, have gone down significantly. So one of the interests in my group is Fusarium head blight of wheat. So shown on the bottom here is some grain with different degrees of damage by the disease. So you can see, a few wrinkles here to something that looks like completely covered in white mycelium from the fungi. And as part of our harvest survey program, we can do track the occurrence of these damaged kernels across the different crop districts. And you can see that this is some of the data here spanning from 2003 to 2019. And then 2016 was an epidemic year. I've heard estimates of $1 billion in damage from Fusarium head blight just in that year alone. So it's definitely a bad year for producers in 2016 for Fusarium head blight. And within my program, we can take a closer look at those Fusarium damaged kernels. We can grow the Fusarium on the Petri dishes. We can look at the fungi under the microscope, and we can look at the differences in the different morphologies of the spores, for example, to try to get an idea of what's causing the disease so that we can better manage it. And this is sort of the traditional microbiology approach to managing a lot of these diseases or monitoring for them, was to do this culturing based techniques. But you can imagine, my eyes aren't that great to begin with, but staring through a microscope for a few hours, you start to go cross-eyed, and trying to, increase the throughput of this is a bit challenging, because there's only so many microscope slides you can stare at before your eyes start going googly, and you have a hard time telling these things apart. So the approaches have changed since the genome of Fusarium became available. So in the mid-2000s, the Fusarium genome was released. And this really unlocked a whole slew of tools for researchers to now better understand these fungi. So this graph here shows the number of papers that have been published on Fusarium graminearum in the literature by scientists after the release of the genome, which is around 2002. You can see that there's a massive throughput from the research community to unlock this knowledge that was captured by the Fusarium genome. And as part of that knowledge, they understood, the toxins that the genes, toxin genes that are in the fungi that are involved in making DON, which is one of the primary concerns with Fusarium, is contamination of toxin with DON. And there's different derivatives of DON. There's 15A DON, 3A DON, and now there's a new variant, NX, that's popping up. And the genetic cause of these different variants of the toxin produced by the fungi are known. So by doing sequencing and gene analysis, they're able to identify this gene Tri8, and differences in this gene give rise to either 15 or 3A DON, and differences in this other gene located somewhere else in the genome, Tri1, gives rise to this NX producing toxin. And doing monitoring of these toxins by chemistry is actually very expensive and challenging to do. But to do a DNA test for these genes is very easy, and they could be used as a proxy to know what toxins we're expecting to see produced by the fungi. So for example, this here is a machine in my lab which we use to do our DNA testing. This is a 384 well plate, and we have this robotic dispenser here. It picks up samples in the plate and it dumps them into this metal chip. And this metal chip is going to have 5,200 little holes in it. And each one of those holes will be a separate DNA test. So we can run many, many tests on each individual Fusarium damaged kernels to be able to identify what the species is, as well as test for those genes and toxins and identify which toxins will be produced. So this could be done at a very low cost and a very high throughput way. So, I was showing you before, we're just scanning microscope slides looking at the different spore morphologies, but here we have data for over 37,000 kernels, which we've tested for different Fusarium species, as well as toxins. And we can see, Fusarium graminearum is the most prevalent species that we're finding, and we're detecting most of them are this 3A DON chemotype, but we're seeing some with this 15A DON chemotype, and rarely do we detect this NX chemotype. But this sort of gives you an idea of how the scales have sort of changed in light of genomics and access to this new information that the genome sequences have brought. And similar to Leanne's issue with the canola pathogens, Fusarium is also a complex disease. You have interactions between the different species, and that can lead to different disease outcomes in producer fields. So monitoring for these different species is important, as is monitoring for the toxins, because even different chemotypes or isolates within the species can actually infect differently based on the toxins that they produce. I'm going to switch gears a little bit from the serial killers, pardon the pun, and talk about the cereals with a focus on wheat. So we also do quite a bit of genomics in crops to develop new tests that we can use to monitor for varieties, because varieties will differ in their quality profiles, and we can do these DNA tests to get an idea of what the variety purity is in different grain samples. A lot of this research is also used by breeders for crop improvement, because they can develop assays or DNA tests to better monitor for traits of interest in their breeding programs to develop superior cultivars for producers. So I'm going to focus more on the crop improvement standpoint. And from crop improvement, producers are mostly interested in yield, yield stability, so protecting yields against losses from bacterial and fungi and insects, as well as resistant to cold and frost, heat and drought. This is particularly important in light of climate change. And then also the marketability of the grain. We want it to look good. We want it to have, bake a good loaf of bread, right? So to give you an example of some of the traits that we've looked at through genomics, Fusarium heblite resistance, resistance to the DON toxin, quality, we've looked at yellow pigment, which is an important quality of pasta and durum wheat. For quality, we've looked at grain cadmium accumulation, the genes responsible for that, as well as some physiological traits in the plant. So a solid stem, so some stems of wheat are sort of like a tree trunk, and some of them are more like a drinking straw. They have a hollow stem. And this solid stem, hollow stem trait is important for resistance to the wheat stem sawfly and can provide nutrients during periods of stress as well, and also provide support during grain filling. So I'm going to talk a little bit more about these traits in a few minutes. But before I do that, I'll talk about some of the work that we've been doing that's been supported by the Canadian Producer Commissions, as well as Genome Prairie and Genome Canada. And these are the 10 Plus Genomes Project and 4D Wheat, where we're really trying to capture the diversity of wheat so that we can better understand the genes responsible for these in-demand traits. So we use a tiered approach where we're capturing a lot of information about a select few individuals, and then we sort of gather a medium amount of information on a fewer lines, and then we've got lots of information lots of lines with a little bit of information, and we can use this database collectively to capture the diversity across wheat, not just in Canada, but around the world. So the 10 Plus Wheat Genomes Project sequenced the ones highlighted here, which are from different breeding programs around the world, and we can capture the information of some medium level information of some genomes, as well as use lower end sequencing information to impute the genomes for thousands of individuals. So when we looked at our genome data, we found some really astonishing differences between the genomes. So this is an example of one of the major differences that we found, where there is a huge chromosome arm that just sort of got swapped between two chromosomes in one of the wheat lines. So we were able to capture just a lot of this really interesting variation that's important for different traits. So this one here is likely associated with some rust breeding that was done in the past. And we developed databases to make the data more accessible to breeders and non-experts, so that they can take full advantage of this sequencing information. So we have these web-based browsers, and you can visualize all the information quite easily. We developed quite a few of these different tools. So here's some videos of them here. So you can interact with them, select individual chromosomes, look at this is the chromosome swap in a linear view and a circular view. This one here captures different haplotypes, so regions of the genome that are from different areas, from different sources. And all this information can be integrated into breeding programs. So to give you an idea of how that actually works in practice, the breeding and research are now fully integrated with one another. So from a breeding perspective, you're interested in selecting parents, developing germplasm, then you're selecting for individuals in your breeding program that you've made crosses with, and then you're evaluating the genes in those materials. And this sort of feeds back into the research where we're evaluating if those genes are working in those systems, which new genes are available in existing germplasm databases, and can this information be used to select new parents that can then feed back into the breeding cycle. So this really gives us full comprehensive understanding of the diversity available to breeders, as well as applying that diversity in applied ways to improve crops, and then feeding that back into research with it, which then can help guide the next round of breeding. So to give you a couple of examples of how we've used this data in applied breeding, one of the traits that we looked at is this solid stem trait, which gives resistance to the wheat stem sawfly shown here. And the gene from sequencing, we're using Oxford nanopore long reads, we're able to identify this gene, which is present in multiple copies in solid stem wheats that are resistant to the insect. And then we can develop a high throughput quantitative PCR test to be able to identify if there's multiple copies of this gene, And when we do that, we find that it correlates perfectly with whether or not the stem will be solid or not. So now breeders can just take the information from this gene, this DNA test, and they can use that to quickly assess whether or not their plant is expected to give a solid stem or a hollow stem. Likewise, for the grain cadmium, we were able to use the sequencing information of high and low grain cadmium accumulators to identify a 17 base pair duplication in high grain cadmium accumulators in this one gene, which ends up being a metal transporter gene. And this duplication causes the gene to no longer be functional. It has a premature stop codon, so the gene's broken. And because this transporter of the metal is broken, the grain accumulates the cadmium instead of it being sequestered in the roots. So some wheat cultivars, when you grow them in high cadmium soils, the grain will be the cadmium will be transported to the grain instead of being stored in the roots. And the reason, based on sequencing the genomes, we were able to identify was because of this difference in this one gene. And we can develop just a DNA test based on amplifying the length of that region and see if that duplication is present. So if it's duplicated, the DNA fragment will be larger and it'll have a higher band. And if it's not duplicated, the gene will work and it'll have a lower band. And this is just a really quick DNA test that you can do across many different wheat lines. And it immediately tells you if it's going to accumulate high or low grain cadmium. And it can be used as a proxy, right? And we've tested it across global lines as well as Canadian lines, and it's a perfect predictor of grain cadmium accumulation. And like I said, a lot of these chemistry tests for things like grain cadmium are very expensive to do. So now breeders, they don't have to do the more involved chemical testing when they're developing their lines. All they need to do is run this one DNA marker, and then they're able to assess whether or not it'll be a high grain cadmium accumulator or not. So to sort of summarize what I've shown you before in this talk, genomics really catalogs this global diversity, and it can be used both for pathogen monitoring and crop improvement. It can be used to understand how pathogens in crops work and to discover new genes and DNA markers. And I showed you some of that with the testing of the different chemotypes, toxin types for fusarium, as well as developing DNA tests for wheat breeding for monitoring for the solid stem trait for resistant to the wheat stem sawfly, and also for the grain cadmium accumulation. And really, these tools are all new to these communities, so it's really an exciting time to be applying genomics to agriculture. Yeah, so thank you very much, and look forward to your questions here.
Thanks, Sean. Very interesting. And I had no idea serial killers were actually that innocuous in appearance. I thought they would, wear hockey masks or something. Okay, so we have had a couple questions. I noticed, Leanne, that you did answer a couple of questions that came up specifically about whether or not your services were available to people in Saskatchewan. I don't know if you want to comment on that.
Yeah, so we operate as a testing lab, and the Get Tested program is specific to Manitoba Canola Growers members. So yes, we depending on the year, see samples from Alberta, depending where they are with kochia. We'll see samples from Saskatchewan for pathogens, maybe that we've seen like verticillium that they haven't. So yeah, we're not geographically restricted. So
okay. I see Rupinder has asked, Leanne, what are recent molecular marker techniques your place is using to identify the wild genes having resistance?
Yeah, so think of PSI's role as really at that applied close to the field thing. So we leverage a lot of the work, that Sean and folks in the science community do. So we wait for a marker to be developed, and then we will validate that. So I wouldn't say that we're involved that far upstream, of identifying, resistance genes. Now, if the question is, do we identify a novel source of herbicide resistance, for example, that work is underway. So it's a bit of both, but think of us as very applied. So if a tool is developed, we want to play with it and see if we can actually use it.
Okay. And we're also asked here, how do you inform the end users of the advances that you've highlighted in your talk?
Is that for Sean, maybe?
I think it was actually
Sure.
for, yeah, both probably.
Okay, well, we can both tackle it. We'll tag team that one. And most of the markers that we develop, we use in projects that are actually integrated with those that might be using them. So for example, when we're doing genomics as part of the 4D wheat project, that project has the pathologists, the breeders, everybody is sort of in on the same project and we're all working together on the same team. And then once we complete the project, the data is shared publicly through the literature. It's sort of outlining how the assays and tools work and what we're able to find and how they might be able to be used in an applied context for, either breeding or monitoring for different crops or the pathogens or what have you. So usually the best way to have the data used is to bring everybody on board. And then once it is, we want to make it available, not just to Canadians, but everybody, right? Like we're all on the same team globally. So.
And I'll answer that from our perspective. So every sample that is submitted, the submitter agrees that we can use that information at an aggregate level as part of surveillance efforts. And so what that means is we share it with the province of Manitoba. We share some of the canola, obviously the blackleg results with the Canola Council, and they use it as part of their outreach. For us, it's very important that the grower knows what's going on, but also that the community has this extra information to round out some of the information that either comes out of traditional surveillance or that comes out of labs. One of the things that we have found, and this is a question back for Sean, is timeliness of that turn. So we try to turn our results back to the grower within, 10 days, unless, where we can. With the amaranth and herbicide resistance ones, we try to do those, within 48 hours because it's timely for them to act. One of our challenges has been waiting on the broader surveillance data that seems like it takes a long time just for surveillance data. And then if we have to wait for it to be published, it could really be years, whereas our ability to influence was 2 years ago. So wondering how that, from that sort of research environment, we can speed that process up would be something I'm interested in.
Yeah, that's a good point. And I think that the timeliness of getting that information out there is very important. I would say with our harvest survey program, where we work directly with producers to provide them feedback on their grain, we have a service standard. I can't remember exactly what it is. It may be 10 days upon receipt of the sample to do a full workup of the quality and unofficial grade, and then report back to the producer because they need that information now to make decisions about what they're going to be doing with their grain. For our survey work, where we do sort of these broader analysis across, all the different crop regions. So for example, I gave a snapshot of that heat map where there's the fusarium across years. So that's a report that goes onto the CGC website every year, shortly after the last few samples trickle in that we're able to include into the survey. We compile the data and we put it on our website so that it can be used right away by our industry partners and those that might be interested in it. And we're always working to better provide information that might be useful to people. So if there is information that people are particularly interested in, we often get requests for information to be able to help support specific questions. And then those we do sort of on an ad hoc basis so that we can ensure that people get exactly what they need. But we try to make those general ones go out, shortly after the last samples come in so that they can be available to everybody.
We actually have until 12:30, so we have plenty of time because we've got some questions here that people have been asking. Do you think advantages of gene editing on wheat could include improving productivity and increasing quantities?
Maybe I'll take that one and I'll start by maybe taking my Government of Canada hat off and speaking just as a general scientist. I'd say the GMO conversation is a complex one. So I think that the industry needs to sort of be careful when they decide which direction they would like to go in and around GMOs, and we certainly monitor for those in the Grain Commission. With respect to wheat, I think that, you know, you could see gains from things like CRISPR and genetic modification. And in Canada, the way that our regulatory system works with the Plants with Novel Traits Act is we don't necessarily regulate the process by which the plant was generated, but we regulate what it is. And that's very different than what's done internationally. But if we're hoping to export our grain to other countries, it's important to understand how those other countries are going to receive and perceive anything that might be modified by CRISPR. And then that would probably be the major hurdle, I guess, for anything related to CRISPR getting off the ground in a crop such as wheat. So the short answer is, sure, CRISPR could probably make wheat better, but in a practical standpoint, due to, you know, export and who we're hoping to sell our grains to, it might not be a wise decision, at least in the short term, unless anything changes in how the global markets sort of perceive these technologies. And I'll put my Government of Canada hat back on.
And I'll mention, again, certainly speaking only on behalf of the science and the tool, I think more importantly, the tool. I think all the tools need to get a good kick around to see what the potential is. You know, the shame would be not looking into validating or advancing a tool, at least into a test environment, before we decide we can't use it because of. And that, to me, I think would stifle the innovation that we've seen. Whether it gets deployed or not is almost a second conversation compared to what could be the benefit. And not to get controversial, but I was just, you know, not sure how far behind I am on my reading, but Argentina, I believe, has, you know, a drought-resistant wheat trait, right? That was brought in through genetic modification. In a world consumed by drought right now, that may stop without, you know, an evaluation of where it could fit. Seems like we're not using the tool that we could. Doesn't mean we could or should, but to not even, you know, entertain the conversation, I think is the tragedy that could happen. So from our perspective, all technologies should be looked at. That doesn't mean they all have to be deployed. So.
And on that note, we do have a question of how does drought, how is it affecting drought resistance in crops? Do we have specific projects for drought resistance at molecular level study?
Yeah, so I'd say, you know, across all crops, there's researchers in Canada, both at the university and in government, that are working on all the top priority traits that are important to producers, including drought. And I think in the recent rounds of government mandates and funding, there's an increased priority that's being placed on climate change and being able to respond appropriately to the needs of producers in respect to the climate variability that's expected for producers in the future. So the short answer is yes, and probably that research is diverse, you know, across all crops, and you know, even in Canada, where we, you know, have our breeding programs, you know, they're in diverse regions. Some of them are very drought, known for their drought, so then they can test the varieties and evaluate them under those conditions and select for, you know, the best crops to respond to those needs.
I have a question on that one too, probably maybe for Sean or some of the other folks that may do more breeding than I do. This year, at least in Manitoba and most of the prairies, we saw not just drought, you know, so water deficit, we saw heat stress at key stages. And they can look the same, you know, reduced yield, shitty looking plant, right? But they're really different physiological mechanisms. So the degree to which there might be some genetic tools that we could use to parse out what is a drought response and how to deal with it, and what is a heat response, which, you know, at a certain stage of flowering or pre-flowering could be devastating, and we may miss it. So I'm intrigued by how we can take kind of plant physiology and apply some better tools than just observation, because I don't think we can parse that out just with observational studies. So I don't know, Sean, if you're aware of any kind of, physiological work like that?
Yeah, so I think, we're starting to look less at, organisms of having individual genes and really being a system of complex genes that are all interacting. They're biological systems, right? And many of the different genes are interacting, which makes some of the traits very difficult to parse. But so, for example, the solid stem trait is actually an example of a trait that's, thought to be giving resistance to drought and providing nutrients in the stem that can be remobilized during periods of stress. And we identified a causal gene for that using genomics. And now we have assays that we can monitor for that in breeding programs so that they could be applied in an applied sense to develop superior cultivars. So that's, an example right out of my presentation today of how that information can be used, right? And as part of that research, the gene responsible is actually called a transcription factor. And these are genes that regulate other genes inside the organisms that are doing a whole slew of different things related to, drought, stress, in controlling the reproduction within the stem. So, they're not just, one gene doing one thing, they're sometimes controlling many different things simultaneously. And, with the genomes, we can now understand how these systems work to try to optimize the systems, for Canadian producers. So you could use, like an entire systems approach to try to improving the crop as opposed to, trying to tackle these things one at a time.
So now we have a question specifically for Leanne. Most companies who want to adopt genomic sequencing into their service are deterred by cost and unsure that the investment may be worthwhile. How was PSI's experience in the challenges and rewards of genomics adoption into your processes?
Great question. And I think Sean had the setup slide for my answer, which is the cost has, become almost de minimis. We actually spend a lot of money on assays, on assay reagents and equipment to do traditional things. We haven't found that adopting the DNA-based testing, once you've invested in the equipment and the skilled training, is that much different. The sequencing, we're just at I would call it our infancy on that, and understanding what the tools are. And that's part of our involvement with SeekNet, is really how accessible is an iSeq with a cartridge thing that costs $1,000, and my God, we don't want to ruin it because we'll spin the wheel on $1,000. That for a small lab like ours is very important. But we're also lucky in Canada that kind of discovery work, because we're a private company, is scientific research and experimental tax credit eligible. So we do have a buffer that that allows us to at least manage some of those costs. So it's mixed. I would say that the costs of entry are probably high to buy qPCR and some of the support equipment, but after that the ongoing operational costs are probably lower than I thought, and we expect those to continue to come down.
There's another question. Are there genomics derived assays or tests today that can provide growers confidence in their selection of varieties to address adverse weather events?
I'd say the variety registration system has, the seed guide. I don't think warm weather and drought leading to harvest is in the seed guide, unfortunately, and it's hard to assess for every possible scenario. But there are varieties that are known to provide some tolerance. I guess I can't really answer that question directly, other than if that information is important to you, then there may be ways to try to capture that information. But right now, I don't think that's part of the captured well, maybe in that process, or maybe to the level that's desired. And
I'll add on to that too. So we've seen some of that, and trying to use the word proxy. So the soil zones really are proxies for that kind of climate-related adaptation. So a variety well suited to brown soil, for example, compared to something in the Red River Valley, not just soil type, but then fusarium pressure and things like that. So we have the broad adapted, varieties, and then you have these more regional approaches, and that's where you know, some of the programs that we see, whether it's, you know, in Manitoba, it's the McVet program, right, that puts varieties out in our regions so that we can see the differences against the standards. I don't think there's any substitute for that, because lab experience validated in the real world is I think what we're trying to mesh. So to rely on a lab method, absent a real world, you know, sort of connection, I think would probably not be as satisfying as one would want.
Let's say that the current system, you know, does evaluate varieties across a number of different sites with different soil types. But you know, every farm is different, so it's hard to, you know, cater that seed guide directly to your farm. And you know, that's when some on-farm research is likely needed to make sure you get it right. It's definitely not an easy thing to juggle, especially this year. So hats off to all the producers for managing the challenges of this year and that drought leading to harvest.
Yeah, definitely a challenge. I've been asked about SeekNet doing mobile services. We're actually not currently doing mobile services, although we are definitely encouraging more actually localized testing, like in farm, if people can actually do that kind of testing rather than having to take it into a lab. We're definitely wanting to encourage that process.
And well, maybe Raquel, one of the tools that we've got for that is within the SeekNet family it's called a bento lab, a little bento box. It's basically a portable, you know, little PCR with, and you can run a gel. And we're interested in that kind of tool because, again, you would take a portion of the lab to the field for a yes, no, maybe a tissue test. Is it robust enough? Will it hold up? You know, I mean, can you read the thing in the sunshine? You know, these are all the practical pieces that we like, you know, to experiment with. So I think some of the tools are there. It's, you know, are they practical in the way they are? I know one thing that we would love to find is a faster, easier way to extract DNA from soil in the field, because collecting a soil sample, shipping a soil sample, and then us extracting the lab is probably the biggest time and money you know, investment that we make. So, and for Clubroot, for example, 70% of the samples we test are non-detectable. So we're bringing 100% of the samples in for 30% that have quantifiable DNA.
Yeah, it's surprising that really the problem is the sample prep.
From a sequencing perspective, you know, the technologies are developing very quick. Every year is different. So now, for example, Nanopore has technologies that around $100 per sample to run, and they can work, you know, off of USB, through USB devices. So they can work off of computers out in the field, and people are using it to do field-based sequencing. But, you know, the data analysis and, like you said, the preparation of the DNA for sequencing might be a little bit more involved for you know, a generalist who's just hoping to go out in their field and get some information right away. But I wouldn't be surprised if, you know, a few years down the road that the you know, the rate that the technologies are changing, that some of this would be even more accessible than it is today.
And on that, we have found that again, the matrix is what's important. You know, so insect vector disease, like squash a bug, see if it's carrying, you know, a pathogen, you know, aster yellows or something like that. That, you know, insect vectored, those ones are easy to sort of smash and test, you know, really quick. Then comes tissue, right? Because it's green, there's ample amounts of DNA. And then you've got soil or grain, right, where it's dry, you know, you have to manipulate the matrix a little bit more. So it really depends on the type of sample as well. So the, you know, we'll probably get the easy ones done first, and then hope for advancement on the challenging ones.
We have another question here that harkens back to your mentioning that Canada has a different approach in regulating gene editing, which is a product-based approach to the PNT regulation. How do you think that the current regulation in Canada affects breeding? Is it a burden on breeders and wheat producers? What prevents farmers and producers today from adopting gene-edited seeds?
Yeah, I guess that's
kind of touched on that.
Yeah, like the Plant with Novel Traits Act, my understanding of is, you know, they're regulating the product, not the process. So for example, if you generate a crop through CRISPR that has a transgene in it, like let's say a gene from another organism, that might be treated differently than, let's say, deleting a gene from within the plant, where you're not introducing anything new, you're just sort of tinkering around with a gene that's already present. So they're regulating what it is, not the process. And I think the largest challenge with that system, I think it's a very innovative and intuitive system. I think that system makes sense. And I think that Canada is taking a very forward-thinking approach and science-based approach with that PANT with Novel Traits Act. But it is different than how other countries are treating it, where they're regulating the process, whether it be CRISPR or introducing genes through genetic modification through other means. And I think that challenge from an import-export standpoint is where that is going to create the biggest challenges. From a producer and breeder standpoint, there's going to be a lot of research going to be going on regardless. Science community is always sort of ready to adopt, but also ready to accommodate what the industry needs, right? And right now, if it's not needed, you're probably not going to see it being released. But when the need pops up, I'm sure that the breeders will be ready.
Okay. Leanne, you mentioned a whole farm approach with synergies between crops and their pathogens. Can you talk a little bit more about that?
Yeah, and again, this, it's as much sort of looking to the vision, but it really starts with, when we look at, for example, canola pathogens, in an ideal, recipe formula, a grower is only growing canola 1 in every 4 years. Those interim 3 years, something else is being grown on that land. We know from traditional crop rotation studies that the preceding crop can influence either, nutrient use or pathogen or weed presence. And so that's really the basis of it, is taking what would be a traditional agronomic approach to understanding whole farm and seeing if, with the DNA tools that we've got, is if we can actually get better answers than, this crop before that crop means you may have X outcome. Can you make it Y outcome by understanding better if you've, I use the example of blackleg. If we can shift the races so that we're preserving our major gene resistance longer because of the interim crop, that could be a good thing. But is there also something in the natural growing environment that we could leverage? So if I knew I had X, somethings, right, of a particular substance that would promote growth, that we can, measure or monitor, maybe I can manage fertilizer use. Maybe water optimization is different when there's this. We just, I don't know, but I would think until you open your mind to the possibility of getting out of this sort of crop pathogen silo or, single and look whole farm, we're not really going to answer a whole farm economy type of question.
That's an interesting thought. Yeah.
It's complex as heck, right? I mean, but as Sean said, we've got tools that 3 years ago we didn't think were affordable, and now our little lab in South Winnipeg has got a sequencer, right, that we think could become part of just when we ran this sample, and now we're going to run these through just to see how different they are or not different. It's kind of exciting, but you have to have a philosophy that says that is something that's desirable, and that's how working with the growers has allowed us to ask those kind of questions. Don't have the answers yet, but we're at least open to the question.
Okay, and last but not least, because I think we're getting towards the end, how can producers access some of these resources and testing that you guys have talked about today?
Yeah, well, I guess with the Grain Commission, we do have the Harvest Survey program, so I encourage all producers to sign up. If you send your samples to us as part of that program, you get a standard set of tests that are done on those. But you can never hesitate to reach out to myself or to the Grain Commission, and we can do our best to support you if you have a specific question or need that's come up on your farm. We often have the resources either in-house or we're connected within the industry to point you in the right direction to get you what you need. And sometimes, that's getting you in touch with people like Leanne, right? Because that's what her entire office is there to do to provide that level of support for producers.
Right. And I'll mention that too. So from PSI's perspective, on our website, like everything is driven through the website because we need positive permission to test. So every assay that we offer a commercial quote unquote test on is on the website. But we also take a lot of questions, mostly from, I would call, industry groups that hear from their growers about an emerging problem. So in the soils, for example, we're doing a lot of work with potatoes. Manitoba is expanding pea acreage, so some of the soil-borne pathogens, again, they're very similar in how one would run the assay, just the marker changes. So those are the platforms we can look at. So I call it soil-borne. We're interested if it affects Manitoba. Aerial pathogens, obviously. Insect vector, I'd love to do more on. For example, aster yellows impacts our carrot industry as much as it, doesn't impact our canola, but it's the same, it's basically the same organism. So working with the carrot growers, to understand, if the leafhoppers are carrying, the pathogen or not. Those are some of the things that we get involved in that can be done. Doesn't have to be a test because we're trying to determine, is it a problem for Manitoba?
Interesting. Okay, that's good to know. Okay, I think we'll wrap up. It doesn't look like we have any more questions. We've got a few comments in the chat. Oh, Rawa has raised her hand. I'm gonna unmute you, Rawa. I have to. There you go. You can talk now. Rawa is my colleague, Genome 360 project manager in Saskatchewan. I think you can talk. Maybe I haven't unmuted you properly.
Actually, you gave me the opportunity to unmute myself. Thank you.
Oh, okay.
Sean and Leanne, this was an amazing presentation. Thank you. I've organized some of these meetings and I've still learned so many new things. So thank you for your presentation. But I have a question for you, Leanne. It's from a business perspective. So the prairies are not very popular for being quick to adopt technologies. I imagine Manitoba is actually doing better than Saskatchewan, but either way, we're generally a little slower to adopt it. So I was wondering, even though you've shown in your presentations and other presentations that there's a lot of added value in adopting genomic testing, I was wondering, how have you generated interest among growers by adopting this at PSI?
Well, I think some of the quick answer is by addressing a need. So example, for Clubroot, there was no DNA-based testing in Manitoba. So that was obvious, right? You would get no answer. For things like herbicide-resistant kochia, the molecular assay is so easy to do, and it provides such a quick answer that when a grower is faced with that issue and wants to confirm that they have a problem, it's very easy for them to connect with that. So I think it's really needs driven. I think part of the challenge for general surveillance is the immediacy of the problem. And then equally important is coupling it to a solution. You remember in my presentation, I talked about to us, it doesn't go far enough just to provide a result. You got 10 million spores, have a nice life. That doesn't help me as a grower, right? I need to know, well, what does that mean? So give me context and give me a solution. And if the solution is don't grow canola, okay, I'm maybe not listening to that solution. But if the solution says maybe grow canola with this, mix of, major genes or something, now you've empowered a decision. And I think really that is the part that we went in with that mandate. So it's been easy to meet that mandate.
I see. Thank you.
Okay. I see Rupinder also had her hand up. Did you want to unmute yourself?
Hi, thanks for this amazing information. Shan, it was really nice to hear you after like almost 2 years. And Leanne, your presentation was excellent. And my question is here, how we can aware the farmers about the genome testing, especially for the variety evaluation and seed purity. Do we have any extension programs so that we can aware at the farmer levels? The industries, they are very much aware about this technology. So what we can do at the farmer level so that they become part of this amazing project?
Yes, that's a great question. And, like Leanne said, a lot of it's driven based on need. And we do get requests from producers and agrologists or consultants that work with producers trying to, solve specific issues around variety purity, or we work directly with industry partners who are receiving grain and require some additional testing to be done. And a lot of this just happens behind the scenes to ensure that the industry is, working as it should, as a quality check sort of within the grain handling system. I guess if there is a need by producers to have some of this testing done, which does pop up, we do get requests from them. This can be done by the commission, but there's also seed companies that do testing as well, and we share a lot of our testing methods and technologies with these testing companies, so that they could work more closely with producers to provide those services if those services are needed by those producers.
The one aspect I'll raise, and I mentioned it in canola, is that, so there is a raft of information that the breeding community and seed companies have on what the genetic makeup of their variety is. So resistant to fusarium, okay, but Sean, a whole lot of work went into that sentence, right? Whether it's specific major genes or a combination of, deployment of minor genes. In canola, we have voluntary adherence to providing information at least on the major gene on the bag, so that a grower can take the genetic testing information and actually use it to choose a variety. That's not universal, and I'm not aware, and somebody can hopefully correct me if I've missed one, outside of canola, I don't know that anybody talks about the major genes that they're deploying. So without that piece, the ability to take an action for the grower is non-existent. So I think there more transparency either from the regulation side of it, so that there is disclosure of, what is the genetic makeup in that sack, would really help advance genetic testing. In Australia, I think for canola, it's mandatory that the major genes for blackleg, for example, are on the bag. Those are the same companies that we have in Western Canada, where it's a voluntary system. So I'm all for disclosure, right? We have a tool, but we have an opaque box of how that tool could be deployed, absent what is actually in the varieties.
Okay.
I'd say from the wheat perspective too, within the seed guides, they do split up the different types of resistance, for example, to fusarium versus resistance to DON. And for many of the cultivars, they do have variety reports about the specific varieties. And, the breeders will put out, full reports on the genetics behind them if they carry specific disease resistance genes against different races. And this information is also applied by the seed companies that distribute to the producers. So for example, if wheat's resistant to the orange wheat blossom midge and it has the SM1 gene, it's actually sold as a blend to help protect the resistance gene against breakdown. So a lot of this is sort of behind the scenes. I'm not sure how interested producers are in the specific genetics behind it, but the information is usually public for all the varieties. So the breeders can access it the producers can access it if they need to. And if they have trouble, they can always come find me. I'll help find you what you need.
So that's that. It's almost like bringing those two data sets together, really to support the decision making, which is what I think is at the root of all of our interest in testing anyway. So
we're having some great discussion, but it looks like we're out of time. So obviously we need to have another one of these webinars, and you'll be hearing from me. I want to thank Sean and Leanne both for presenting, and our audience for being so intent and interested in participating. So thank you all, and you will probably be hearing about another webinar from us soon.
Thanks
Talk to you later.
To SeekNet for having us share this kind of cool work that's being done.
Yeah, thank you so much for the invitation. Nice to talk to you all today.
Okay, bye.
Bye now.